Good morning, Dr. Roddenberry coming to you live from Willow Springs, North Carolina. How are you doing today? Hope you had a great weekend. See, we've got a couple of people in the audience. Why don't you go ahead and type me a hello in the chat bar. Let me know who you are. I'll bet Ronique is out there because Ronique tried to come to my 9 o'clock class this morning. That's great, Ronique. I love your effort, bud. Hi, Britt. Good to see you. Good morning, Shamay. Good to see you. Fantastic. All right. So who we got? Katie Bauer. Good to see you. Anybody ever? Does anybody ever seen someone with Down syndrome before? Anybody know anything about Down syndrome? If you're out there in the chat world, why don't you go ahead and tell me, what do you know about Down syndrome? Ever met somebody with Down syndrome? What causes Down syndrome? I'd be curious to see what you know before we get started. What do you know? Okay, let's see what we got here. I know somebody's typing in the chat bar and can tell me. So while you're typing, I'm going to go ahead and switch right here. All right. So, Oh, well, I'm going to carry on. Maybe somebody will type in the chat bar. Good morning, Imani. How are you doing? All right, let's go ahead and get started if we can anyhow. Maybe somebody will tell me what they know about Down syndrome in the meantime. It's caused, there we go, it's caused by an extra copy of the 21st chromosome. Yes, it's called trisomy 21. Trisomy. You're only supposed to have 2 chromosomes. You've known some people with Down syndrome. I know it has to do with their chromosomes. Yes. I used to volunteer in the Miracle League Baseball League, helping the kids play baseball. And a lot of the kids had Down syndrome. It's typically going to be associated with some facial deformations, moderate to more severe levels of intellectual deficiency. And a lot of Down syndrome kids actually aren't verbal either. Yep, fantastic. But what I wanted you to know, what to tell me was that it's a chromosome disorder. So today we're going to be talking about the genetic perspective in developmental psychology. Now this is, as I suggested to you, developmental psychology is a multidisciplinary view. Today I'm going to talk about what the biologists and the geneticists would have to say about the developmental process. Because if you think about it, your development starts with your set of instructions, and your genes are the set of instructions designed to create you. Now, these genes are subject to evolutionary pressures. Some genes are selected for and some are selected against. These genes then work through a special unfolding process, which may or may not be affected by the environment, we'll call epigenetics. And then when we talk about abilities and disabilities, they can also be traced to malformations in these instructions. So the developmental process all starts with the story of genetics. Now, keep in mind, I'm a psychologist and not a behavioral geneticist, not a geneticist. So I'm going to do my best here, but don't ask me any super hard questions today because you just don't know if I'll be able to answer them. Okay. Good morning, Ronique. Glad to see you here. Got a great class today. Hi, Andrea. I see we have Juanita here. Katie, thanks for your comments. So let's talk briefly now. Why do we care about genes? Now, there's a perspective that is listed in chapter 1. One perspective on evolution, on developmental psychology, is that our qualities, our abilities, our genetic instructions are driven by a selection process, which selects for some genes and against other genes. I don't know if you folks know anything about natural selection. It was first sort of elucidated by a fellow named Charles Darwin, and he talks about the intergenerational changes, morphological body changes that and behavioral changes that occur as a result of competition for scarce resources. Okay, actually, I'm getting a note. Britt Boyd's has a bad connection. Is that me or is that you? My confidence monitor that I'm looking at, my monitor suggests I'm coming through pretty clear for everybody, so it might be you, Britt. If somebody else is having problems, go ahead and type in the chat bar. But let's talk a little bit about natural selection as a theory. So it suggests that any organism creates too many offspring that cannot survive. These offspring are somehow different. Some have big beaks, some have short beaks. Some are tall, some are short. Some are skinny, some are heavier. Some have big wings, some have small wings. It's all sort of random variation. And these species, there are too many of these species, but they compete for enough food to survive, for enough water to survive, but there's not enough. So only those that are best equipped for that world will get the resources that they need. And these animals that get the resources will grow to maturity, will have babies that are more like them. So natural selection suggests that the qualities that we have are decided upon, and the reason that we have the heads that we have such big brains, that we have thumbs, that we have what we do, is that these changes in our body, this body that we have, is best suited for survival in our environment. Now, this is regards morphological change, why the brains look the way they look, why the way the hearts look the way they do, why the thumbs look the way they look. But you know what? There's also a branch of evolutionary theory called evolutionary psychology, which does the same thing with regard to behaviors. So, why do parents and their offspring have such close relationships? Why does that behavior occur? And evolutionary psychologists would say the same forces that shape our bodies also shape instinctual behaviors that we are born with. So when we get to the chapter on socio-emotional development in infancy, I'm going to revisit this term attachment, which I'm sure all of you heard in introductory psychology class. And we're going to talk about attachment as an evolutionarily adaptive behavior that causes offspring and primary caregivers to bond close together, which provides safety to those children that grow up. All right. And so if you look down here, we have morphological and adaptive behavioral changes that you can see. And the crazy thing is these morphological changes and these behavioral changes sort of co-occur. So if you think about it, humans are born extremely helpless, right? Little human babies are more helpless than most other animals you will meet. And you know what? It's because our brain is so big that if it came complete, we wouldn't be able to be born. We wouldn't be able to make it through the mom's pelvic girdle to be born. So the human brain has evolved. It's going to be big when it's mature, but it's born missing a lot of stuff. And so babies are helpless because they don't have this brain material. So this behavioral change of developing a close relationship with the mother co-evolved so that babies wanting to have these big brains could be taken care of while their brains grew. So from an evolutionary perspective, there are biologists who look at how bodies and brains and behaviors changed and how they can be explained as functions of evolutionary pressures. Now, what the heck does this have to do with genes? Well, genes represent the vehicle. Genes represent the vehicle by which these biological qualities are passed. So your behavior in your body is to a large part determined by the instructions that you have in your genes. The idea behind evolution is that if your body and behavior is successful enough, you will grow to sexual maturity and pass those genes, those instructions on to the next organism. Those changes in your body or your behavior that are not adaptive will either get you killed or will not permit you to engage in sexual reproduction, and your genes won't be passed on. So, the story of developmental psychology, the story of human development, begins in the genes. Now, if you look underneath me, what you're going to see are the karyotypes, the, if you will, chromosome pictures of both a female in pink. I'm going to be gender stereotypical. Exactly. There you go, Ronnie. Be careful of who you have babies with, right? So you'll see on the left, I'm being gender stereotypic here. On the left, we have the pink karyotype of the female, and underneath me, we have the blue karyotype of the a male. And a karyotype is simply just a picture of your 23 chromosomes. And you will see that there are 23 pairs of chromosomes, or 46 total chromosomes. Now, you folks probably learned from biology class that when we engage in sexual reproduction, each gamete, the male sex cell and the female sex cell, each has a half complement of chromosomes. So the female has 23 chromosomes, gamete has 23 chromosomes, the male gamete has 23 chromosomes. When sexual fertilization occurs, you get a cell that has 46 chromosomes, half of those chromosomes coming from mom and half of those chromosomes coming from dad. And this is why Ronique says, be careful of who you have kids with, because your kids are part of your spouse's genetic line, right? Now, just a quick note, the first 20 through 22 pairs of chromosome are not associated with sex. We call them autosomes, and we're just going to differentiate those from the 23rd pair you see down here at the bottom. They are called the sex chromosomes because this is where your biological sex is determined, okay? You'll notice the female has two X-shaped chromosomes down here. Fertilized egg cells that have two X cells in the 23rd pair will be biologically female. And if you'll notice down here, the male 23rd pair has an X chromosome and this Y chromosome. There is a specific gene on the Y chromosome called the SRY gene, the sex determining region of the Y chromosome. And this gene turns your bipotential gonads into testes at about 7 weeks of pregnancy. All right. And so if the little organism does not have the SRY gene, these bipotential gonads will not turn into testes. They will automatically turn into ovaries. And so your sex chromosomes are where your genes are where your sex is determined, right? Now let's talk briefly about what we're talking when we talk about chromosomes. Your chromosomes, you have 46 of them, 23 pairs, are these thread-like structures that are bundles, really tied and convoluted, wrapping structures of this DNA, all right? And DNA, if you remember, is that double helix molecule that contains genes, okay? And genes are the units of hereditary information. Genes make proteins, amino acids. Genes build things and they make things work, okay? So your chromosomes contain DNA. Your DNA contains genes, and your genes are what build you and cause you to run. They're the ones that build and run the system that is you. Now, it turns out that your full genome has about 30,000 genes. You have 30,000 different genes together that create who you are and what you are. Now, you may or may not be surprised to know that you share the exact same genes. 80% of your genes and a mouse's genes are the exact same. You might say, holy cow, we look so different. How can those be the same? Well, if you think about it, you have 4 limbs, a mouse has 4 limbs. You have a head, a mouse has a head. You have ears, a mouse has an ears. Now, those ears may look different. That head may look different. Those limbs may look different. The heart, the stomach may look different. But in a sense, you have a lot of the same general structures that a mouse has. Did you know that I think we share 99% of our genes with apes? And each and every one of you, I believe, if I'm not incorrect, we are 99.9% genetically similar. The similarities among humans far, far outweigh the genetic differences. It's that 0.1% of the genes that create all the variation that you see in human beings. All right? Now, I don't know if you folks have heard, but they have recently mapped out all of the genes in the human genome. This was not possible before high-speed computers, but with the advent of easily available high-speed computers starting in the 80s and 90s, it was possible to actually map all the genes in the human genome. Actually, I will tell you, probably 15 or 20 years ago, the belief was that there were over 100,000 genes in the human genome. It's not until recently that we've really been able to come even to a pretty good idea of how many genes are contained in a human genome. And the Human Genome Project has allowed us to map all the genes in the human genome to provide opportunities for research and treatment. Yes, we are in the, they are, I think they are at the process where gene therapy, I think, which is what you're talking about, Ronique, gene therapy is now becoming possible. I'm not sure how far along they are in using gene therapy, but I know that it is now a discussion that they are happening. Now, the crazy thing is, gene sequencing used to take a long, long time. Now they can do it super duper quickly. And the neat thing is, they actually, in treating cancer now, they can go in and look at the genetic structure of your cancer to find out if the tumors that you have are more susceptible, and they have different medicines, and depending upon the genetic structure of your cancer, they can actually find they can use different drugs that are more effective on the particular type of cancer that you have. I have a friend going through lung cancer right now, and that's what's happening in his case. They were able to sequence the genome of the cancerous tumor, find out if it had a mutation, and this mutation allowed them to pick a more effective drug to treat his chemotherapy drug to treat his cancer. Now, what we also do is people are looking for potential disorders that you may be born with, that risks that you may have later in life. And this genome-wide association method and linkage analysis are two techniques where we can sort of begin to identify the markers and even the specific genes that will tell us if people are at risk for this disorder or other disorders. Now, these disorders aren't as simple as just one gene or another, so these particular methods are still in their infancies, but genome-wide association and linkage analysis are allowing doctors to, like I said, telescope in or microscope in and start to see if there are specific clues that can tell us whether or not someone is going to be struck down by a particular disorder. And then for research research purposes, there are people who are now sequencing genomes from people around the world to look at genetic variations and maybe even allow us to understand the genetic markers of disorders that strike different populations of people, from Tay-Sachs disorder, which strikes Ashkenazic Jews, to sickle cell anemia, which attacks mainly people of African and Sephardic descent. And so there are lots of things that we're doing now that we're able to understand, go in and actually look at these set of instructions that people are born with, right? Now, just keep in mind, this is probably a review, but you get a set of instructions from your mom and your instruction for your set of dad. Now, the reason we do this is so that we can combine and begin to create different variations on the human being to see what kinds of strengths we can have. Also, if your parent has a gene that is damaging to you, your other parent has an exact replica. So you get two sets of genes that describe how to build your nose, build your ears, build your chin, build your heart. And so sometimes one parent may give you a set of instructions that contain a genetic mutation that is damaging. As long as you've got the gene to build that structure right, what you're going to find is that you, in fact, turn out healthy instead of having the mutation caused by the deficient gene. Now, because we get the exact copy of instructions from our mom and the exact copy of instructions from our dad, we have two sets of instructions for how to build us. Those full instructions are called your genotype. But as the organism is built, it may get brown hair from its mom or blonde hair from its dad. It may grow up to be tall like the mom's side of its family, or it may be short like the dad's side of its family. The resulting morphological structure that occurs out of these dual instructions is called your phenotype. Your phenotype, okay? Right? So, in a sense, let me see, maybe I don't know if I can write this properly. So you get hair color gene from mom. Let's say that's a brown hair. And you get a hair color gene from dad. And let's say that says to be blonde. Okay, so B is brown and B is blonde. Now the question is, this is your oh my goodness, what am I doing here? Excuse me. This is your genotype. But what color will your hair turn out to be? Your genotype says brown and blonde. Well, which is it going to be? What color will your hair turn out to be? Well, in this case, your hair will probably be brown. Okay, now let's go back to our chart for just a minute. Now, why does this happen? Your mom and your dad each give you a set of instructions. Those set of instructions may agree. Mom says blonde, dad says blonde. You're going to be blonde. But what if your mom's gene says you should have brown hair and your dad's hair says you should have blonde hair? What's going to happen is your body is going to decide, your genetic material is going to cause you to grow one of those two colors. Now, there are several principles we can use to understand how genes, genotypes, come together to create what you actually look like, which is your phenotype. All right. Now, the first is that in the case of simple gene expressions like maybe your hair color, some genes are typically seen as being dominant rather than recessive. So if you get a gene for for brown hair and a gene for blonde hair from each parent, the brown hair is what we call the dominant gene. So anytime it's paired with a blonde gene, you're going to turn out to be a brown haired person. Now, what this means is that people who have a non dominant phenotype, like blonde hair, it means that they were given a blonde gene by both their mom and a blonde gene by their dad. Now, I'm using hair because that's what's on my mind right now, but we could talk about hair color, we could talk about skin color, we could talk about muscle definition. Any of these you'll get a trait for building it from your mom and your dad. And sometimes some traits are dominant over others. So if we get 2 copies of each trait, the dominant genes prevents the expression of the recessive gene. 75% brown. There you go. Listen to these people. I have some nurses in here. Kaylee Morgan saying 75% brown. Okay, Kaylee knows what's going on. Nicole, red hair is a mutation. Okay, Nicole must be redheaded and also a medical student there. All right, now, here's the thing. I don't know if you know this, but it is not as simple as one gene versus another. Did you know there are actually 6 genetic spots that are responsible for the full unfolding of your sexuality? Yes, the SRY gene makes you a male, but there are actually 6 different spots that contribute to the full expression of your sexuality. So it's not just one gene, sometimes it's a combination of genes that work together to create different aspects of that final thing. So being male isn't just, or female isn't just one thing. It's 4 or 5 different genes working together in your body. Okay. Now there are sex-linked genes. So there are some genes that fall on the 23rd pair of chromosomes. And what this means is that if a mother receives a mutation trait that is recessive, like being bald-headed, she probably has a dominant trait on the other X chromosome, which will balance that out. And what you're going to see is that females hardly ever develop what we call pattern baldness. They don't go bald up here because baldness is a sex-linked trait that occurs on the X chromosome. Now, if men have the baldness trait on their X chromosome in the 23rd pair, they don't have another X chromosome to override that recessive baldness trait. And so what you're going to see is that men are much more commonly bald than are females. And that is because pattern baldness is a sex-linked gene. All right. And then sometimes this more bizarre phenomenon is that sometimes a gene will be more or less likely to be expressed depending upon which parent gave it to you. So it may be a gene for hand length on your 16th gene, and if you get it from your dad, it might be more like a mutation for hands might occur on the 16th chromosome. But if you get that that chromosome from your dad as opposed to your mom, it may manifest differently. Okay? Ha, funny. Funny, 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 Ronique. Okay? Now, some disorders. Now, why are we interested in all of this? We're interested in this as developmental psychologists, primarily because we're interested in some of these disorders. What genetic concept represents the 6 genes of sex? This idea of polygenetic inheritance. So one of the things, if you know today, there are people who argue about whether or not there's 2 sexes or whether or not there are intersexed people. And actually both, in a sense, are right. Genetically speaking, you either have the Y chromosome or you don't have the Y chromosome. You're either genetically male or genetically female. However, mature biological sexuality occurs in the development of many sex-related organs in both our primary sex characteristics, so the growth of a penis or a vagina, the growth of testes uh, or ovaries, the growth of secondary sex characteristics, breasts for females, the change in body shape for both males and females. And so this cascade of changes, sometimes there will be genes that don't work right, and this cascade of changes won't occur. So the X and Y just sort of start the process, but there are other genes that are responsible for creating the hormone testosterone or estrogen that cause different brain biological structures to grow that then contribute to the process of sexual maturation. I know, right, Katie? It is kind of confusing to sort all of this stuff out. It certainly goes beyond my mind or comprehension. Uh, Now, the reason we're interested in this, one of the things that developmental psychologists do, you inherit a rare disease from only one parent. My daughter has a rare genetic disease inherited from her dad. Wow. That's interesting. We might ask the question, uh, why inherited from her dad? Maybe it's a sex-related disorder. And so she got an X, wait a minute, let's see, hold on. She got the Y chromosome from your dad. She got the X chromosome from you. Maybe from her dad. So maybe it's the, I'm not sure, maybe it's the fact that the genetic disorder was carried on the Y chromosome, the male chromosome. So it was something that your genes couldn't override. So the dad had it on that Y gene. It goes into the 23rd pair. You don't have a corresponding Y gene. So that disorder is uh, expressed. Her dad and from his mother. Okay, you know what? That might be what we call genetic imprinting. Maybe it's a gene that responded differently because it was passed on to you by your dad's side than by your side. Okay, dad gives them to daughter and moms to boys. Okay, Imani, now you're asking an even harder question. You're killing me here. Imani, every time I answer it, she just keeps making the question a little bit harder for me. I give up. You win. I'd have to sit down and map that one out for about 10 minutes to understand. So the dad gives it to the daughter, and the mom gives it to the boys. That sounds like genetic imprinting, honestly, the expression of a gene differing depending on whether or not the gene is passed on by the mother or the father and whether or not it goes to a daughter or a son. That's very interesting, though. Okay, so again, we're interested in abnormalities that affect the developmental process. And as I suggested to you, developmental psychology is interested in helping humans live longer and better lives. That starts by understanding the disorders that children are born with. And so some of these disorders are related to larger structural abnormalities of the chromosomes or missing or extra chromosomes. And then some of these disorders are related to characteristic mutations that actually occur within the chromosome. So you folks, if you look over here on the left, you'll see some common chromosomal syndromes. Down syndrome is the extra chromosome in the 21st pair of chromosomes. A lot of chromosome malfunctions like this will result in terminated pregnancies. Down syndrome is one of a minority of actual numerical aberrations in chromosomes that actually results in a live birth. A lot of these, if you're missing chromosomes or have too many chromosomes in a lot of places, it may lead the pregnancy to be terminated. But for some children, they're born with this 47 chromosomes And this chromosome leads to moderate to severe intellectual disability. And like I said, the facial deformations. And also, in my experience, many of the Down syndrome kids were not high functioning enough to even have language. You know, it turns out that Down syndrome is much more likely in mothers over the age of 35. So if you're a mother who gets pregnant over the age of 35, you are considered a high risk pregnancy, and more invasive evaluations will be done of the baby because they have a higher likelihood of developing Down syndrome. Now, I've also got here means that people aren't born. No, no, they are not, Britt. There are some people, like I said, there aren't that many. Trisomy 21 is one of the few times that you'll have 47 chromosomes and still go all the way to pregnancy. A lot of them aren't. A lot of them will lead to termination of the pregnancy. That's why you'll have that miscarriage early in the pregnancy period. The genetic material isn't right, and the baby will be terminated on its own. On its own. But yes, here's the crazy thing. Here's the crazy thing. There's this term called mosaicism. It turns out that even people with Down syndrome, not all of their cells have 47 chromosomes. So they actually, if you look, it's kind of like a mosaic. Some of their cells will have 47 chromosomes, and the more cells they have that have 47 chromosomes, the more severe the disorder is. So the bizarre thing is, even these people, not all of their cells are creating this, are generating this 47th chromosome. I can't tell you any more than that. If you want to learn about mosaicism, you need to go read it on yourself, on your own. But yes, Britt, it is quite possible. Now, you're actually going to see some people with 47 chromosomes have that 47th chromosome in the 23rd pair. They have Klinefelter syndrome. These people have two X's and a Y. Now, this is a genetic male, but that extra X chromosome is going to cause them to develop feminine secondary characteristics. I can actually show you that real quick. Here's a person suffering from Klinefelter's syndrome. And what you'll see, you see this male right here, you're going to notice that he has very large fatty deposits in his breast. Now, these actually, he's not just a heavy person. This guy's actually got the genes that help generate a little more fat in this area than will be normal for most of us. And so you'll also see that this guy's shape probably won't be particularly manly, and this fellow may not even be fertile. Now, the weird thing is, these symptoms are sometimes so subtle that people with Klinefelter syndrome don't even figure out that they have this until they're in adulthood and having difficulty getting pregnant. And they may find out then that they actually have this syndrome. Yes, you saw this in gender, right? Absolutely. Absolutely. And then you've got Turner syndrome, which is where you have a genetic female, see, there's no Y, who's actually only has 45 chromosomes. This person doesn't have the 46th X chromosome, but they are a genetic female. And then when you look at somebody with Turner syndrome, what you're going to notice is a genetic female. Notice the broad shoulders, and this female is going to have more of a masculine look. And typically, females with Turner syndrome are super duper short, 5 feet or less, and a lot of times they actually are sterile as well. Exactly, the tomboy look. Now, you've actually got some genetic men who have an extra Y chromosome. Back in the day, they used to think that these men were more likely to be criminals because they had two Ys. turns out these guys are not more likely to engage in criminal behavior. However, they tend to be taller than normal. Okay? Now, sometimes you will get Fragile X syndrome, and this is where the X chromosome in the 23rd pair has abnormalities. Now, for females, it isn't a problem because they have another X chromosome in the 23rd pair, which takes over for the damaged X chromosome. But for your men, they don't have an X chromosome that's healthy to take over for the abnormality in their 23rd X chromosome. And so you're going to see Fragile X syndrome, which also leads to intellectual disability and learning disabilities. It's more common in males than in females. So why do some children develop intellectual problems? Why do some children develop school-related problems? Why do males and females' sexuality not turn out in the prototypical fashion? You can actually trace these back to chromosome or abnormalities that people may have. Now let's contrast that with gene-linked abnormalities. Genes are your instructions, so you may find the proper number of X and Y chromosomes. You might have 46 chromosomes, but if you telescope in on your DNA, on your genome, you may find that there are mutations or changes to specific areas of that genetic instruction. And these genetic abnormalities, mutations, may lead to disorders. Cystic fibrosis, diabetes, hemophilia, Huntington's disease, phenylketonuria, sickle cell anemia, spina bifida, and Tay-Sachs disease. These are all child disorders, congenital disorders that you are born with that are caused by your genes. For the exam, these are a lot of disorders. I will want you to know Down syndrome, Turner syndrome, Klinefelter syndrome, those three. And then I'm going to ask you to know, from this one, phenylketonuria and sickle cell anemia. Okay, those are the genetic disorders that I will ask you about on the test. Phenylketonuria. What they do is they do a little blood test when every baby is born. It's called a heel prick test, and they look at your blood. And what they are looking for is the level of phenylalanine in your blood. Now, some people have a genetic disorder in which their body does not break down phenylalanine. When this happens, these people are diagnosed with phenylketonuria. Yes, it's the newborn screening, absolutely. If your baby has PKU, your baby will be put on a very special diet that they are on for the rest of their life. Because if phenylalanine builds up in your system, it leads to cognitive deficits and intellectual disability, mental retardation. Okay? And so before 50, 75 years ago, children would be born with PKU. We didn't know anything about it. The phenylalanine would build up in their system, and you would have a child that grew up to be intellectually deficit, to have the IQ in the 50s or 60 ranges. And we didn't know why do some children develop intellectual disability like this? We now can screen for that and prevent this intellectual disability from happening to children today, the PKU test. And the second one I want you to know is sickle cell anemia. And this is because I think this is interesting because it does have this ethnic variation that I said we were interested in with the 1,000 Genomes Project and other things. We know that there are genetic mutations that affect certain populations of the world. In Africa, to your Sephardic Jews, and all the other people living in Africa, there is a gene that provides protection from malaria. Unfortunately, the unintended side effect of this gene is it causes the misshape, it causes the red blood cells to become misshapen at times. And this blood disorder, these misshapen red blood cells then don't run through the capillaries properly and decreases the supply of oxygen to the body. It occurs mainly in people of African descent. Like I said, Africans and African Americans and Sephardic Jews, that is, Jews from the northern part of Africa. So on the test, I'll want you to know both of these. Phenylketonuria, because we can now do something about it, and sickle cell anemia, because it represents genetic variation that you'll see in different parts of the world. Okay? More than 7,000 genetic disorders have been identified, and so we could either learn 2 or 50 or all 7,000 of them. For this test, let's just settle on 2, right? Okay, now, that's all I really had to say today. Does anybody have any questions? Please, somebody ask me a question. I insist that you ask me at least one question before we end class. And I'll wait. Awkward silences are okay. That's a great question. You know, I'll bet using what we know about, we could do that if we did. I'm not sure if they've been able to research using the genetic research methods that I mentioned to identify the specific gene yet. I don't know if they've identified the specific gene, Katie. If they have, they could probably test for it to suggest that it is more likely in your infant. I guarantee in the end they still follow through with the blood test, but that's an interesting idea, knowing that we ought to be on the lookout and maybe do the test a few hours earlier for this child than for another child. You know what, Katie? That's a great question. And thank you if you would do that, because I don't know the answer to it. Any other questions? Okay, I'm going to go ahead and log off this lecture. If you have any questions, you can answer. You can send me an email or a text, and I'd be more than happy to answer them. Remember, for this week, we do not have the webinar, but we do have the second part of the discussion board. You need to go in and respond to at least 2 other people's comments to complete the discussion board. If you didn't get any points because you didn't complete last week's discussion board, if you did the webinar, after the final webinar is due, I will add the webinar attendance. So if you didn't do any discussion board, you have a 0. But when I do in the final grading of that Friday morning, I'll put a 2 in your column. If you did the discussion board and came to the webinar, after I finish updating attendance on Friday morning, you'll see a 3 in your column, and you'll get an extra point of credit on your collaboration grade. Okay, but just remember, you have to finish up the second part of your discussion board. Now, if you didn't do anything on the discussion board and didn't come to the webinar last week, you can still earn half credit for the discussion board, but you have to go in and complete the discussion board by creating 1 unique post and then responding to 2 other people. All right, folks, y'all have a great day. I will see you on Thursday. Take care.